For my research paper, my mentor and I decided to research the traversal of or cell migration of circulating tumor cell clusters that contribute to tumor metastasis, with a focus on Sam Au and other researchers in the article Traversal Circulating Tumor Cell Clusters in Capillary-Sized Vessels. I chose this topic because I'm interested in oncological practices, and since artificial intelligence or AI is constantly making new developments. And with the rise of AI, biotechnology will continue to advance and can contribute to filling in some of the gaps in previous studies. I want to not only learn more about the biomechanics behind this study, but to also identify some gaps and open questions that arise within the study. So circulating tumor cell clusters, also known as CTC clusters, are multicellular malignant cancer cells that plant metastasis or the spread of malignant tumor cells to distant organs. These clusters can sometimes contain up to 100 cells and are derived from either primary tumors or from the proliferation of singlets. The clusters then combine with other tumors or tumor or non-tumor cells, such as cancer-associated fibroblasts or epithelial cells, to name a few. It was discovered, as shown in Figure 1 on the slide by Au's study, that CTC clusters can transit through capillary-sized vessels through a specific process. And Au and the other researchers engineered microfluidic constriction devices using 5, 7, and 10 micrometer microchannels to mimic the microcapillaries in order to observe the process of traversal and find the significant impact that CTC clusters bring to metastasis. And in fact, CTC clusters are able to plant metastasis with 50 times greater efficiency than singlets, as shown in this slide. And a study conducted at the Massachusetts General Hospital Cancer Center found that over 90% of observed cancer cell lines in cultured CTC clusters, some containing over 20 cells, traveled through vessels as narrow as 5 micrometers, which helps in identifying cancer signs and therapeutic targets. So I'll be playing this video right here, which shows the process of CTC cluster traversal. With the microfluidic devices that Au and the other researchers used, the study or the video shows that the clumps of tumor cells go through 2 transit phases. The first stage, which is known as the entry, is where the cells come into contact with the walls and reshape into elongated shapes. And the cells that then are no longer deformed, but travel at a constant velocity in the travel phases. Once entering the vessels, clusters reorder into a sequential strand of spherical shapes. In the human body, they pass through the endothelial cell-coated microchannels. However, for the sake of simplicity, the experiment was conducted in non-cell-coated microchannels. Okay. And then for some of the factors that take place, the researchers concluded that the CTC clusters are able to transit to distant organs by forming into single file chains. And in their study, they found that the clusters restructure into single file chain geometries in order to fit inside the narrow vessels, as shown in Figure 3. Once out of the vessels, the cells quickly clump back together. And I realized that since the cluster forms into a long chain, the microfluidic forces can quickly move the cells to distant organs, which supports clusters having 50 times greater efficiency than singlets, which I showed a few slides back. Adhesion or molecular attraction plays another role in the transfiguration of CTC clusters into single line chains and back into clusters. Cell adhesiveness is driven by the extracellular matrix, or the ECM, which consists of a network of secreted extracellular molecules that maintain cell and tissue cohesion and fill in the gaps between cells. And in addition, the strength of adhesion is determined by the number of chemical bonds on a cell surface, meaning that the greater the number of chemical bonds, the stronger is the cell adhesiveness. And figure 4a, like on the top right corner, shows the different adhesive strengths. Strong intercellular adhesion at 75.0 times 10 to the negative fourth joules over meter squared. Moderate intercellular adhesion at 15.0 times 10 to the negative fourth joules over meter squared. And weak intercellular adhesion at 1.5 times 10 to the negative fourth joule per meter squared. This means that with like stronger adhesion, the clusters stay clumped together, but at moderate adhesion, the cells begin to form into a single file chain, which indicates that like at the first or entrance of the capillary constriction, that area has moderate adhesion. And that weak adhesion, that's where the elongated chain leaves the vessel and the clusters begin to cluster back, or the cells begin to cluster back together like they did in the beginning. So it's like a cycle together, and this is shown in figure B. Another factor that plays a role in CTC cluster metastasis is hydrodynamic forces. And as aforementioned, the formation of a long singular chain forms from clusters lowers the resistant force of hydrodynamic forces, possibly causing their potential greater efficiency. And in the graphs shown in this slide, figure 4a illustrates the significant difference in velocities between clusters and singlets when the cluster diameters are equal to the spheres of equivalent volumes as all constituent cells. But contradictingly, in figure 4b shows that not much of a significant difference in velocities between clusters and singlets when the cluster diameters were considered equal to the diameters of their largest constituent cells. And CTC clusters being able to traverse through the vessels were once argued as impractical by previous studies, but it's now been proven that CTC clusters can actually traverse capillary-sized vessels by other means. And in fact, because of their ability to form into single file chains, CTC clusters have 90% more efficiency in spreading cancer cells to distant organs than singlets. And this process of squeezing in as an elongated form is pretty universal among patients with different cancer types, whether it be those with breast cancer or melanoma. And this is supported by patient participant experiments with studies of cell-to-cell interaction energies and in vivo xenotransplantation studies, which are integrated with computational observation. And just like the results showed in a few slides back, CTC cluster transit is supported by adhesion and hydrodynamic forces. But there are some limitations to the study, such as some lack of resources, which led me to come up with some open questions. And I hope that through future research, these areas can have more definite answers. So the first question I have is, how are CTC clusters able to make such drastic changes in shape temporarily? Although it's been concluded that CTC clusters are able to transit by elongated chains, it's unclear as to how they are able to do this in such a short period of time. CTC clusters don't often show up in blood circulation and can essentially be anywhere in the body. And this makes it hard to identify therapeutic targets, which calls for more advanced technology to be able to observe or perform in vivo experiments that can detect and collect CTC clusters. Another important question that I thought of while learning about the hydrodynamic forces and velocities was, do the CTC clusters form in sequential order when elongating into strings? Since adhesion matters in the traversal of CTC clusters, I wondered whether the strength of the molecular bonds in each cell determine the order of the chains. This led me to come up with follow-up questions like, does this have anything to do with hydrodynamic forces and the larger constituent cells on the outer areas of the string of cells, or vice versa? And could it have anything to do with how strong the chemical bonds of the cells are that determine the adhesion? The final question that I came up with the study, and this has to do with like the adhesiveness and molecular bonds, was what are the molecular factors that determine the adhesiveness of CTC clusters? Adhesion has to do with the molecular attraction of cells, and knowing the type of molecules involved with CTC cluster traversal can help with pinpointing targeting areas and developing new interventions. And this particular focus can also dig into the area of cluster formation, which ties into my first question of how its dynamic changes work. So the conclusion shows that the gaps in this study requires more advanced technology to be used. The researchers used microfluidic devices that mimicked microchannels in order to observe how CTC clusters traverse in vessels, but they didn't really explain the dynamic changes, sequential order, and molecular factors. So I think that similar designs to the microfluidic devices that they used can help to identify those specific assets or aspects. And knowing those specific aspects can further produce more coherent and therapeutic results. And some ideas could be by using computational image processing or in vitro experiments. These are my resources, so thank you for your time. And I'd also like to thank my mentor, Mr. Feng, for guiding me throughout my research experience as a part of Talaria Summer Institute 2023. He hasn't only explained the topic to me, but has also encouraged me in my research process, whether it be through good advice and tips. And he's also taught me some formal scientific writing techniques, which I think I really needed. And TSI has been a very valuable experience in engaging with a mentor who really knows a lot about my topic and also gives me the opportunity to learn something that I probably wouldn't have known as much had I not been a part of this opportunity. So I'd like to thank you guys again. Thank you.